Good morning, Scotland. Good morning, Edinburgh. I'm Denis Lafontaine, and you are in session 2 on RNA modification. We have grounds to cover, both in terms of RNA modification and in terms of the biological process they impact. We will first hear on translational recoding from the team of Itau Yu in Rochester. The team had shown previously that if you convert a uridine into a pseudouridine at a stop codon, well, this will lead to the ribosome to read through this stop codon. And today the team has now converted two uridine into pseudouridine, one at a premature termination codon on an mRNA and another on a tRNA at position 36, and they are studying how these pseudo-U's interact with one another, leading to stable interactions that promote ribosomal read-through. We will then move on a case of differential translation from the team of Sherif Abouelela in Sherbrooke, where they are characterizing a particular pseudouridine on the large ribosomal subunit, which is important for the differential translation of mRNAs depending on the size and GC content of their untranslated regions. We will then move to further exploring the potential of nanopore sequencing to map RNA modification, and we will hear from the team of Professor Tsutomu Suzuki in Tokyo that most RNA modification leave a beautiful fingerprint on the sequencing reads, but that for some modification, the fingerprint actually spans multiple residues across the modification, making assignment quite difficult. And in these and other cases, it will be important to extract features directly from the raw signal, and that's what we're going to hear about from the team of Tsutomu. We're going to hear about a signal alignment method which has allowed them to map more than 13 different types of tRNA modification in multiple species. We will then move on M6A, and we'll hear a talk from Julian Koenig in Mainz. And M6A is a modification which shows a distribution biased towards the 3' end of transcripts, but today we will hear about a case where M6A encoding sequences seem to play an important role in a novel decay pathway that requires ongoing translation. We will keep on talking about M6A, but now from an RNA catabolism perspective, and we will hear from the team of Professor Fan-Yan Wei in Tohoku University in Japan, that M6A, which is a rather stable nucleoside that can be sometimes excreted from cells, may act as a signaling molecule. In that case, it will bind transmembrane receptor of the GPCR type, and this will in turn trigger pathological immune response of the type 1 allergy. And we will close the session with a bacterial talk from Joel Belasco's lab in New York, and we will hear about a dinucleoside tetraphosphate molecule, which was known for many years as an alarmone, but which may in fact play many of its role rather as a cap, stabilizing particular transcripts in stress conditions. And today we will hear about two key enzymes involved in decapping this particular cap and processing the uncapped transcripts. And with this, I think we are ready for the session, and we are ready to welcome the first speaker of the day. And I would like to wish you an excellent week in Edinburgh, and I hope I can catch up with you later this week. Bye-bye.